So far, we have talked about the citric acid cycle only as an energy harvesting mechanism. We have seen that dietary carbohydrates and amino acids enter the pathway at various stages and are oxidized to generate NADH and FADH2, which, by means of oxidative phosphorylation, are used to make ATP. However, the role of the citric acid cycle in cellular metabolism involves more than just catabolism. It plays a key role in anabolism or biosynthesis as well. This figure shows the central role of glycolysis and the citric acid cycle as energy harvesting reactions, as well as their role as a source of biosynthetic precursors. As you may already suspect from the fact that amino acids can be converted into citric acid cycle intermediates, these same citric acid cycle intermediates can also be used as starting materials for the synthesis of amino acids. Oxaloacetate provides the carbon skeleton for the one-step synthesis of the amino acid aspartate by the transamination reaction. Aside from providing aspartate for protein synthesis, this reaction provides aspartate for the urea cycle as well. Alpha-ketoglutarate serves as the starting carbon chain for a family of amino acids including glutamate, glutamine, proline, and arginine. Glutamate is especially important because it serves as the donor of the alpha amino group of almost all other amino acids. It is synthesized from ammonium and alpha-ketoglutarate in a reaction mediated by glutamate dehydrogenase. This is the reverse of the reaction we saw earlier. In this case, the coenzyme that serves as the reducing agent is NADPH. Examination of this figure reveals that serine, glycine, and cysteine are all derived from 3-phosphoglycerate. Alanine is synthesized from pyruvate, and tyrosine is produced from phosphoenolpyruvate and the 4-carbon sugar erythrose 4-phosphate, which in turn is synthesized from glucose 6-phosphate in the pentose phosphate pathway. In addition to the amino acid precursors, glycolysis and the citric acid cycle also provide precursors for lipids and the nitrogenous bases required to make DNA, the molecule that carries the genetic information. They also generate precursors for heme, the prosthetic group that is required for hemoglobin, myoglobin, and cytochromes. Clearly, the reactions of glycolysis and the citric acid cycle are central to both anabolic and catabolic cellular activities. Metabolic pathways that function in both anabolism and catabolism are called amphibolic pathways. Consider for a moment the difficulties that the dual nature of these pathways could present to the cell. When the cell is actively growing, there is a great demand for biosynthetic precursors to build new cell structures. A close look at this figure shows us that the periods of active cell growth and biosynthesis may deplete the supply of citric acid cycle intermediates. The problem is, the processes of growth and biosynthesis also require a great deal of ATP. The solution to this problem is to have an alternative pathway for oxaloacetate synthesis that can produce enough oxaloacetate to supply the anabolic and catabolic requirements of the cell. Although bacteria and plants have several mechanisms, the only way that mammalian cells can produce more oxaloacetate is by the carboxylation of pyruvate, a reaction that is also important in gluconeogenesis.